So I just started a new job at a local startup. So that's why it says former up here. But everything I'm going to be telling you about today, I did in Darwin Sai's lab in cell and developmental biology. So I did my PhD in in biophysics, but in a CDB lab. And then I've been working there for 2.5 years after my PhD. So everything you'll be hearing about today was done, sort of in that period, during my PhD and then that little bit after. Could we tweak the microphone down a little bit? It's just like it's like echoing my own voice back at me. Um, yeah, so today I'll be telling you about a project that I worked on during that time to create tools for dealing with petabyte scale microscope images. So both collecting them and also like doing data analysis on them. So I know that this is a little bit of a diversion from what a lot of these sort of core bioinformatics talks are about. But it's hard to have one without the other, right? It's hard to have the tool without the data. So that's kind of how I split this out. So I'll start out with a couple of introduction slides and then talk, one section about each of those there. And I think
slide.
Oh, there it is. Yeah, so in Darwin's lab, really broadly, the question that's being studied is one of neuroscience. So, to kind of very oversimplify it, right? It's, you have complex organisms. Like here I have a picture of a mouse because a lot of the work in the lab is done on mice, but obviously the same principles apply to any organism with a brain. And there's this question of you have this brain, and if you think about it like a black box, think about it like a computer, it can take in all these different types of inputs, auditory, visual, touch. It goes into the black box, and you get out, an answer or some sort of interaction with the outside world that could be planning, motor control, learning, all these complex behaviors, anything that a human, an animal can do, right? Very, very broad description there. But ultimately, and this has been changing over the last couple of years, but there's still a lot of questions about how you have 71 million different individual neurons inside of the mouse brain. How do they all work together to actually accomplish a goal, right? And this question is becoming even more relevant in more recent years because people are trying to understand this further for machine learning applications, for instance, right? So a lot of the newest sort of visual transformer models are modeled after how we think that the visual cortices in the mammalian brain like operate. And so there is both interest in understanding these questions from a philosophical sense, but also from a applied sense as well. And so, we study on this. We focus on the study of what's called connectomics. So, how do the connections between different neurons, different types of neurons, how do their interactions, sort of create this emergent behavior, right? And I just have two, sort of pictures here. Over here, you have a picture of, sort of mesoscale connectivity. So this is a type of data called DTI. And it's looking at how different regions of the human brain are connected together through an MRI image. And over here, this is just one diagram from one paper, but it's depicting how these different cell types depicted by different colors connect together and create sort of a excitatory inhibitory network, right? And so ultimately, what a lot of our lab is focusing on doing is trying to derive these types of network maps through different methods. So the approach that we use is primarily a light microscopy based approach. Now I just flashed these up, two papers from other labs just to show kind of a small survey of the field. But broadly, what we're trying to do here is create, like I was just saying, these networks, but applied to sort of a whole organ scale. And so each one of these different colors in this diagram up at the top represents an individual cell, and we're trying to reconstruct these at the scale of an entire brain, which is the outline you see there. Now, despite that, the most common approaches for connectomics right now that have been sort of successful at capturing many neurons in the same sample have been using these techniques that use electron microscopy, right? So the idea here is you take a piece of tissue, you freeze it down, you treat it for electron microscopy, then you take an image, you cut it, you take an image, you cut it, you repeat that process. through that, there have been many successful studies who have reconstructed very dense networks where here, these little things that look like balloons, each one of those is actually an individual cell of the Drosophila brain that is being represented there. And then up here, this is a piece of somatosensory cortex in the mouse brain. And you can tell from that, sort of cube projection that it's very, very dense. You can see like all of the cells that are in that volume, right? But the challenge there is that in electron microscopy, the largest experiment that's been done, which was, it was a huge feat, but the largest experiment that's been done is using is this experiment that happened at Harvard that they call the human H01 data set. And in this experiment, they captured 1.4 petabytes of data and reconstructed this big chunk of a human brain. But ultimately, because of sort of the limitations on speed of electron microscopy, they were only able to do about 1 millimeter cubed, right? Which is really small. And I have a sort of scale for you here in a second. And so this sort of cutting edge work that took years of work from like a whole team of scientists, they created beautiful results and all, but it's really unscalable because it's hard to go past that sort of even millimeter scale at Harvard working for years, throwing an entire team of scientists at it sort of scale these days. So, to put this into perspective, this 1 millimeter cubed is what I just showed you. Granted, that experiment was of a human brain. So here's a human brain for scale, and here is 1 millimeter cubed, just to give you an idea of kind of like the difference in scale that you're trying to branch. And then even if you look at a mouse brain is still 500 micron cubed, so or a millimeter cubed. So that's still 500 times larger than what I just showed you, right? And that's quite a challenge. So in Dahlon's lab, we focus on, like I said, doing this using light microscopy-based approaches. So primarily, and I won't be talking about the technique as much today, but using this technique that we call brainbow or bitbow. So the idea here is that in the little animation that you're seeing here, all of these little colored lines, and a lot of them on the projector look a little bit pale, but they all are different colors, if you trust me there. All of those individual cells in the fly brain are colored with different colors of fluorescent proteins. And then we create like a 3D image of them using different microscopy systems and reconstruct them bioinformatically to create these like 3D cell structures of all of the cells in that neural network. So in this case, we are labeling all of the what are called serotonergic neurons in the fly brain. And there really aren't that many in one brain. But this is essentially a picture of what we're trying to accomplish there. Just to give another example from a different paper, this is a neural network that we reconstructed from the mouse basolateral amygdala. So it's just a specific region in the mouse brain. And so again, we reconstruct this really dense 3D neural network of all of the cells that express a protein called parvalbumin or PV. Which you may have seen if you've read any like neuroscience single cell papers. And here we're coloring them based off of cell type just to show that we can sort of discriminate them. But the key thing here is that we're able to reconstruct this actual neural network rather than sort of an artificial neural network.
Just a quick question. Are these images that you are showing us coming from one image from a slide that may have different colors to it because it's looking at different pathways, or are these stitched together images from multiple slides?
So they are stitched together in like multiple dimensions. So it's kind of like you have your microscope, and first you kind of go like this, you scan in X and Y, and then you shift everything up a little bit. Then you take another image in X and Y, and then you repeat the process, right? Okay. And then all of those individual images, right? Because you can imagine there's like a bunch of little squares that get collected. All of those get stitched together, right?
Oh, I see.
Yes.
How do you know you did it right?
That's a great question. The answer is mostly qualitative. If you look at the data and you have a neuron, and it kind of goes like this, and then you reach the edge of a tile and it goes like this and it breaks, that tells you did it wrong. So all of the stitching approaches are basically you shift everything around until mathematically these two things line up, and then you say that's correct. But I mean, that is a really hard optimization problem, especially when, samples get bent in weird ways and stuff like that. So dealing with that is quite a challenge, actually. Yeah, and so to kind of show you what some of the raw data looks like here, this is one of the slides that makes up a figure from one of our papers. Now this one is only a single Z slice. So again, you kind of went along and you could see like a little line right here. That's where it got stitched together, and it's just not perfectly stitched. It's hard to see because it's sort of really dark. But this is sort of what the raw data looks like. But now the challenge here is that if we use the same imaging approaches that I showed you in those two figures just now, if you scale those up to try to capture a whole mouse brain, it's not as bad as the math that I showed you for electron microscopy or sort of the scale there. But there's still experiments that would take they would take days or hundreds of days for like something that's a millimeter cubed, and they would take years for something that's a whole mouse brain. So it's a very similar issue. So during my PhD and after, I approached this through basically two different sides, right? So one is, how can we build a microscope that would allow us to do these images really quickly, right? And then there's the joint problem, which is once you have created this microscope, it's throwing out data. How do you deal with it in such a way that you can achieve it within like a PhD time rather than a lifetime, right? So through this, kind of going back to the theme of the talk, I have sort of 4 tools, if you want to call them that I'll be talking about. In the first section, it's this thing that we call the parallel line scanning confocal microscope, and then that's a lot of words, but I'll talk about it in like one slide. And then the other part, in the other section, it's 3 software tools that we call SysF, SysF-CDN, and InTracer2. So we'll talk about that when we get there, but I just wanted to kind of structure that in your brain. So in this first part, I'll talk about the microscope that I just mentioned, and in addition to Darwin, obviously, this was work with Ye Li and Hehai Jiang, who is a grad student at Purdue. Ye is a research investigator now at Michigan. And then Meng was Hehai's PhD advisor at Purdue. So the microscope that I've alluded to, the parallel line scanning confocal microscope, is a quite complex design that achieves what's called line confocal microscopy. So what we're doing here is, if you've ever seen a diagram of how a confocal microscope works, you shoot a laser, it goes into your sample, it bounces back up, and then you filter the beam out through a pinhole so that you get a very fine Z resolution. With the parallel line scanning confocal microscope, we're doing the same thing, but with a whole line of pixels at once. So the physics is the same. It's just instead of a pinhole, you're sending your light through to like a slit, like a mechanical slit. The physics isn't very important, but essentially it allows you to capture images that are very high resolution, 185 nanometer sort of optical resolution in the sort of X dimension. And it allows you to do it really quickly. So when it's running full speed, it captures about 200 terabytes of imaging data per day, compared to a traditional confocal where the best ones, you're talking, less than a terabyte a day if it's running like full speed, and it's a very expensive sort of model there. So, the principle here, and I won't beleaguer this as much because normally I'm talking more to people who do or who focus on microscopy, but the idea here is that it has multiple cameras. Each camera is collecting a different part of the image at one time. And so when you watch it, there's a bunch of lasers that kind of go like this. Synchronized with that, all of the cameras are capturing. So you're able to collect a lot of imaging data at once because you have 3 cameras running at once, and they're all moving very quickly. That's the summarized version. And so, it allows us to collect images that look something like this. And while this is very chaotic, I can promise you that there's a bunch of neurons in this little panel here. And then, you repeat this process like we were talking about earlier. You scan in Z and you can reconstruct 3D volumes very, very quickly. So, just because I think it's cool, this is what it looks like. It's a big metal board with a bunch of pieces attached to it. It's called a breadboard. And essentially, you have three different optical paths that are bouncing through this diagram here, where you have different lasers. They shoot down. They go through all of these lenses, bounce out, and then it goes out of an objective down here. And then on the return side, it comes up. It gets split out by a bunch of dichroic mirrors, which are mirrors that reject part of the spectrum, but allow certain parts through them to go to our three different cameras. So effectively, it's like having three microscopes running on the same sample at once, purely just in the name of throughput. So, one of the parts of this that is really important is that I sort of custom built all of the controller software for it. And so this is just a picture of one piece of it that we call Cameraman, and now we have a new version called Cameraman 2. So it's a C++ Linux application. It's kind of ugly, but the key thing here is that this is actually running it with 7 cameras attached to the computer. So each one of these cameras is, 3,200 by 3,200 pixels. So this is pushing, thousands and thousands of pixels through the screen at once. It's all GPU accelerated with live image compression, so that as the cameras, collect individual images, it compresses them down and allows us to sort of churn through experiments as fast as possible. So to deal with the onslaught of data, we developed this thing that we call the SNDIF, or Scalable Network Distributed Image Acquisition Framework. The name's a little bit of a nightmare there. And the idea here is that we've kind of taken all of the steps that would normally happen inside of a microscope. If you go to the imaging core somewhere in the med school, and you rent a microscope for an hour, the software that you control there, whether it's the Zeiss software or what have you, will be doing a bunch of different tasks all inside of one desktop application. But when you have one desktop application like that, we found that it was a bottleneck for running these experiments at full speed. So we created this framework that we call SNDIF that separates all of the responsibilities into multiple different parts so that we can spread it out over a network and deal with them all sort of one at a time, and leverage the fact that Michigan already has a lot of supercomputing and network resources that are not necessarily underutilized, but underutilized in experimental science, you just say. So the idea here is that you have all of your electronics that are controlling the experiment, and that's sort of happening externally. And then you have all of the cameras that make up your microscope. Each one is capturing a one frame at a time. Those different frames go into one computer that sort of is running the Cameraman software that I just showed you a moment ago. And each one has a bunch of compression worker threads that are sitting there and collecting up the data, compressing it, collecting it, compressing it, that sort of thing. It then gets pushed out to a separate controller application that's running on a server inside of our building's data closet, so that we have a fiber optic link going between there and our lab. So the images get collected, they get compressed down, they get shoved out into the network to this storage server. From there, they kind of get cached and then eventually pushed to Great Lakes, where on Great Lakes, we do all of the sort of post-processing tasks that have to be done. So, there's a lot of, sort of image deskewing, image flattening, and, sort of normalization that has to occur before the data goes from like its raw state to something you can actually look at and sort of analyze. And then from there, there's a downstream pipeline, which is the second half of my talk. So I won't go into that too far. Oh, and this is basically just a summary of what I was just saying. And the key thing here is that through this pipeline, we can essentially run the microscope at full duty cycle for hours and days at a time. Our longest experiment has been like 4 days, I think. So it allows us to collect hundreds of terabytes of data in a single imaging session, which to our knowledge is not something that anyone else has been able to do without basically having many imaging computers all imaging at once, essentially. So to give you an idea of what this first part of my talk all put together looks like, like I was saying, we loaded up the instrument, and I have this picture just because it looks cool. You can see here this kind of red, yellow, bluish sort of thing. those are our 3 laser beams that are scanning back and forth inside the sample to do the confocal imaging. So this is what it looks like while it's actually imaging, and it's very bright because it's, very bright lasers. And then once you get the data into the server, sort of put it all together, it looks something like this, right? So this is a whole slice of half of a mouse brain. So if you see here, this is a mouse brain. If you cut it, down the middle, this is kind of the shape you get. And this data set is 295 terabytes in its raw state. And because we're using expansion microscopy to make the sample physically bigger, it represents centimeters of imaging space, but each pixel is like 100 nanometers. So those are the kind of 2 scales here, is that you have centimeter size image, but each pixel is nanometer size, or 100 nanometer size, excuse me. So because it's like hard to look at just a single image there, oh, my right hand animation. Oh, I guess, oh, I could kind of play through it by hand, but PowerPoint doesn't want to play it. Yeah, so this is what the data set looks like if you scan through it. The thing that's really cool to look at is up in the top right-hand corner, in the sort of cortex region, you'll see sort of the projections of a lot of cells kind of go like this.
If your pixels are as small as you say,
Mhm.
and the time it takes to scan these runs hours to days,
Mhm.
how do you know that the actual sample you're scanning hasn't moved physically and given you?
So that is both a great question and a great challenge. So the answer is that for these really long experiments, we use, I forget what the compound is, but there's it's kind of like it's kind of like super glue, but it's like biological super glue. You stick it, it's like polyol-lysine. It's a polyol-lysine mixed with something else. You stick it onto the glass, and then when you put the tissue down, it like sticks it down almost like a glue. Like you can turn it upside down and it won't fall off. And then on top of that, we put like a little like acrylic box around it. So we put like this acrylic box around it with a glass cover slide on top, and then we kind of sandwich it all down with glue. So that's basically the answer, is you try to physically stabilize it as much as possible, and then once it's in that state, you can actually pick it up and go like this, and it won't move at all. But that took a lot of trial and error to find one that was that stable. Yeah. Yeah, and so then I just showed you an animation of what it looks like, sort of at the whole slide level. But the key thing is that you can zoom in on any one region, and you actually have like synapse level resolution of all of these cells, right? And so here in red you've got the individual nuclei, and then in green, I didn't say it, but this is a mouse line called GFP-M. So it's a cell line that has a bunch of neurons that are just kind of randomly labeled with GFP, which is like a jellyfish green protein, which is why we kind of plotted in green. This is just another view of what I just showed you, just because like with the green and the red, it kind of gets a little bit overwhelming. But here you can really see that pattern I was talking about of all of these cells sort of projecting in the cortex, and really appreciate sort of the level of detail that you get there. So with this data, you're able to reconstruct 3D neural morphology, just like I was showing you earlier. This is just an example of like reconstructing 3 cells that are all sort of touching each other. So we have 3 cells that are GFP, and then these red ones, these are, I didn't mention it, but they were in the blue channel in the previous one. It's a protein called GFAP that labels astrocytes, so like immune cells in the mouse brain. And you can start to do this level of single cell neuron reconstruction, but on a sample that was collected at like organ scale, right? Which is the key thing there. Yeah. So to demonstrate what this can be done, or what we can do with this, sort of bioinformatically, the nuclei channel, so the DRAC5 channel that was in that image, we trained and ran a NN-UNet model on the raw imaging data to do segmentation of the individual nuclei. So here are the nuclei. This is showing you kind of what the cell, or what the segmentation mask looks like. And then using these segmentation masks, we ran a bunch of quantification on their 3D structure, 3D shape. And using that, we actually performed, sort of like a pseudo single cell experiment, where here or in the next slide, we performed a UMAP and clustering on the morphologies of all of these segmentations. And then this is plotting out the different clusters that we identified. So we split it out into 8 clusters, semi-arbitrarily. And you can start to see how the different layers, say, of the cortex actually sort of are enriched for individual or specific cell types. And here's just plotting the same thing, but just showing like segmentation volume, so like the size of the shell of each cell's nucleus. And this is what it looks like, splitting out each cluster independently, and you can kind of a little bit better see these sort of layer specific structures that I was just talking about, and how like layer cluster 5 is enriched in the striatum there. And so in this way, it kind of it allows you to do kind of something similar to what people are trying to accomplish with spatial transcriptomics data, but this is both 3D because it's 500 micron deep tissue, but also centimeters wide. This is just again my ooh. Oh. Interesting. Well, this was another pretty picture I was going to show you, but I don't think PowerPoint's going to allow me to play it. That's okay. This was trying to show using the system for some of those rainbow images that I showed you, or I mentioned at the beginning. But I guess it's frozen. Imagine seeing a bunch of cells through here that are all different colors, and it's very pretty. But I apologize, it's not allowing me to play it. So that was the sort of second experiment that I was going to talk about. The third experiment here is using the PLSCM system for collecting images of what's called a licon tissue sample. So this was a paper that was in I forgot to put the citation, but it was in Nature a couple months ago as like I think the cover article. Essentially, what they did is they created a technique using NHS esters, which stain every protein that's in the brain. So you soak it, and basically everything in the brain lights up in a specific way. And using this, you can actually create data that's very similar to the electron microscopy data that I showed you earlier. And so in this experiment, we collected an image that's 8.5 terabytes, which compared to the other ones in this experiment aren't very big. But if you were to try to do this with an electron microscopy-based approach, it would be a months-long experiment, and we're able to accomplish it in about an hour. So as these methods continue to develop, we expect that this to be a sort of viable approach to collecting those types of data sets. This video decides to work. So this is what that data set looks like if you scan through Z. So if you kind of look at any one location, you can see these sort of spindly things that go through the image. Those are your individual neurons. And there is work that is being done to train models that are built for analyzing electron microscopy data on this data. So the idea is that it could be a plausible replacement for electron microscopy in these types of experiments. Yeah, and this is just sort of an advertisement of what the work that's been going on the last year and a half or so while this paper's been under review. But we've actually developed a new system that instead of 3 cameras uses 7 cameras to sort of split the spectrum even further. So you both get a higher resolution because the cameras are bigger, but also you get more channels because there's 7 cameras. And so all the stuff that I just showed you is in a preprint that you could read right now. This is coming soon. So if you're interested in this idea, you check out our current preprint, and then hopefully in the next little bit you'll be able to see data from this system. Yeah. So pause right there just for a second. If anyone has any questions about sort of that first half, I'd be happy to.
Very interesting talk. And I was just curious if for the software, is it the only dependency the camera APIs, or if I have if for example a different microscope setup with the software or the pipelines to apply for it, or
Yeah, so the primary, there's a bunch of dependencies just, because it uses the sort of Linux base libraries and the networking libraries and stuff. But other than, the sort of standard C++ libraries, the only dependencies are the camera control library. So version 1 was written with the Hamamatsu control framework, whatever you want to call it, that is used for Hamamatsu cameras. And then the second version, the PVCam, besides supporting more cameras, also the reason we had to rebuild it was because we were restructuring it for the API. It's called PVCam, that's used for kinetics and the higher throughput Teledyne photometrics cameras. And so that's one dependency. The other dependency is just the compression libraries. But again, those are just sort of semi-standard things.
So like the spectrum that each camera captures hypothetically is all like customizable, and then like sort of the script you guys are running on the cluster will handle that.
Well, so so the thing that you're picturing is probably like if you've used a confocal where you have like the selection of sort of spectral range, that is accomplished by using like a prism and sort of splitting everything out, and then selecting a region of that spectra to sort of collect back. For our system, since we're using fixed dichroics and fixed filters, we actually can't change it. So if we want to change it, we have to go in and physically unscrew the filters and like replace them that way. But if so, like with this system, if you tell me you want to image this, this, and this, you can just turn the other ones off.
Oh.
So effectively, that's how we are able to somewhat select the range, but it's not as easily configurable as like a traditional confocal where a lot of them have sort of spectral detectors that can be sort of retuned on the fly. Yeah.
Thank you so much.
Sure thing.
If somebody wanted to get the system working in their own lab, what are some of the key hurdles? Is it like the cost of the detectors? Does it take a lot of knowledge setting the system up? Like what's sort of the key?
Yeah, so so aligning it and actually physically building it is challenging. It's sort of the level of project that if you had a a senior grad student who's experienced in optics, it's not that bad. But if it was if you don't have someone in your lab who is like an optics person, it would be pretty rough to even just get the alignment down with the lasers. And obviously, there's a lot of safety concerns there because these lasers are some of them are multi-watt, so thousands of milliwatts. So that's something, obviously when you're aligning it, you don't run it at that power, but that's something you don't want to play with, safety wise. So that's one challenge, but it's not too bad. The cost is very high, but it is price competitive with like a decent commercial confocal system. So I think the first version, the total bill of materials, granted, purchased during before COVID, so pricing has on everything gone up. It was in like the $250,000 range, I think, plus or minus a little bit. But that is basically the same as like a nice confocal from any of the big manufacturers. Yeah, the hardest part about it is really just the day-to-day maintenance that's required compared to a commercial system. So commercial systems are usually engineered in a way that you can sort of have it installed and let it run for 10 years. And if you don't like spill anything on it, it'll probably survive. This requires a lot of upkeep of like the mirror alignment and that sort of thing, just because of it being an open system, right? So everything's exposed to the air, even just like we did one experiment where if in BSRB, which is where we used to be located, during the summer, they had been cutting off the AC at night to save money, right? Which is, reasonable. But there was actually a measurable change in the resolution of the system over time, where if you plotted it out, when everyone got in the morning and they triggered the AC thing at like 7 a.m., the resolution would increase. And then you would go through the day, it would hit like 6 p.m. and they would start turning everything off, and you would see the resolution like drop down by like 50 nanometers or something, just because of the heat expansion and whatnot of the room as everything went on. So even stuff as subtle as that, when you're talking about these level of resolutions, level of speed, can start to be like actually measurable factors, which makes our life kind of a nightmare sometimes. So did that answer everything? Or sorry, I think okay. Okay, so for the second half, I'll be talking about cloud-based approaches, cloud loosely used here, server-based approaches for managing and analysis of this data using software tools that we've built. And so especially like some of the front end stuff, I was really helped out with two of my former undergrad students. One who's now he was at Netflix, but he told me he just switched. And Menelik, I believe, went to work at Epic, the healthcare company. So they were two software engineering students who during COVID helped me with a lot of the stuff. So for storing our images, as they, I kind of glanced over this earlier, but as it comes onto our servers, we actually convert it into a custom format. So the reason for this is mostly historical, because at the time, a lot of the tools for these for big array manipulations didn't really exist in the form that we needed them to for our use. Nowadays, if we were to re-engineer this, we'd probably use something like ZAR, honestly. But just historically, a lot of our systems still use this custom format that we developed called SysF, or Scalable Image Storage Format. The idea here is that it is similar to a lot of matrix libraries, numerical algebra libraries that you've probably interacted with, but it allows us to take our images, which if you imagine is just a really big array, 3D array, or 4D including color, take it and physically split it up into a bunch of blocks, right? Which is a pretty standard approach. If you've ever used an HDF5 file or a ZAR file, they all do something similar to that. But then in addition to that, we wanted the second layer of indexing where you take each individual chunk and further cut it down. Now, the reason for this is that if this is, just to use the example, 300 terabyte image, if you cut it up into a bunch of chunks that are, each, 1/1000th of the original data set, you're still talking about something that's hundreds of gigabytes, right? So you have a choice of either making these chunks really, really big and having a small number of them, relatively speaking, or making them really, really small and making it really hard to, sort of perform projections and such, where your data on disk will be physically separated. If that doesn't make sense in your brain, write out the math, and you'll kind of see why. If you have a really big matrix and you want to do a single slice of it, it requires that you sort of jump around in the chunk a lot. Now, the challenge there is that if you're compressing your data, you can't randomly jump around in that chunk, right? So we introduced the second level of slicing, where each of these chunks gets further cut down into really tiny chunks, like 32 pixels, so that we have kind of the best of both worlds as much as we can achieve it. There's also some metadata stuff that we're doing to allow us to perform stitching in real time, but I won't really talk about that much. But essentially, each one of these chunks can also have sort of an overlap region that gets cut out dynamically by all of our libraries. And so really the big advantage here is that it is essentially the same speed as accessing something like ZAR when you benchmark it. But if it's stored on a fast media like SSDs, but if you're storing it on hard drives, or even further, if you're storing it on something like turbo storage for the supercomputer, you get a really big uplift in performance with really small chunk sizes, which for us was really important. Yeah, and so we've built, sort of a couple of bioinformatics pipelines centered around this. And the key thing is that each of these chunks don't really interact with the other, so they're, sort of trivially parallel. So we can launch supercomputer jobs where each one of these chunks is being treated separately. And through that, we actually can tap out some of the storage systems on the supercomputer, because it's essentially bandwidth limited. And so this was a screenshot that one of the supercomputer admins that I keep in touch with sent me, where I had fired off a job and we hit 20 gigabytes per second for a couple of seconds, but also maintained a rate of 125 gigabit per second across a chunk of the supercomputer, which allowed us to do a 8 terabyte data set in about 8 minutes, which is kind of fun. So we've done some studies on sort of the best approach, because I didn't mention it. These individual chunks, these little ones, are compressed, and for all of our published stuff, we use a variety of lossless compression methods, we did, some screening of different compression algorithms that perform best on our data and some data from collaborators' labs and such, and essentially ended up with a BLOSC Z standard compression. Those are just two algorithms. BLOSC is a method for sort of cutting it up individual or reshuffling individual chunks, whereas Z standard is the actual compression method. Shuffle means that you are byte shuffling, or is it bit shuffling? Or byte, yeah, it's byte shuffling, which means that for each pixel value, you actually rotate or like kind of transpose it. The reason is that if you have imaging data and you take an image of like a dark room, which is what fluorescent microscopy kind of acts like, most of the pixel values are going to be really, really low, right? If you can sort of imagine that in your head. And that means that if you look at the actual values on disk, a lot of them have the higher bit range as zeros. So if you transpose it, you end up with a bunch of zeros all in a row. That makes compression operate a little bit faster. And so we use this for all of our data. So those data sets that I was saying, or like the one that was 295 terabytes, that actually becomes closer to 100 terabytes on disk because just this level of compression drops it down that much. So this is how we sort of afford storing these data sets somewhat, is applying compression at a first level.
Can I ask a question about that? Since you're doing both lossy and lossless,
yes,
there's bitback encoding from like Qualcomm that you can do a lossless on top of the lossy. You guys looked into that?
So the last section of my talk will be talking about more of the lossy compression stuff. We haven't used that algorithm specifically. What we and so you had sharp eyes. You noticed that it said lossy here. The algorithm that we're mostly using here is a modification of AV1 actually. So it's a video compression method. So imagine you basically take each of these cubes and you treat it like the Z dimension is a time dimension. You compress it as a video chunk essentially. And then when you look back, it's like playing a bunch of videos all at once and then kind of slicing it that way. So that's mostly then the approach that we've taken there. Yeah. We did experiment with some of those more general purpose lossy compression algorithms though, although I don't have any slides about it. Yeah. Yeah, so once you have your data in this format and it's compressed, you start to have a question of like, how do I actually access it, right? So you have a 100 terabyte data set, you upload it to Great Lakes. That doesn't really do anyone any good if you can't like view it, manipulate it and that sort of thing, right? So we have built a piece of software that we call the SysF CDN. So CDN is Content Delivery Network. It's kind of a term used with sort of internet providers. And the reason we call it that is we have servers running on sort of our supercomputer, our local compute cluster, and some collaborators' labs, so that we can kind of grab data from any of them at any time. And the idea here is that it hosts out on the internet API endpoints that replicate different data formats. So the primary one is what's called the NeuralGlancer pre-computed format. NeuralGlancer is a data storage or a data viewer made by Google. They have a division that does electron microscopy based neuron reconstruction studies. So we mostly replicate that API, but we also have a couple of others that we support. And the idea here is that you essentially access the data as though it's uncompressed. The server takes all the data on disk, decompresses it, stitches it back together, and sends you the result, right? So you treat it like it's an uncompressed data set, but the server is essentially acting as a translation layer between the compressed and the uncompressed data. And we have further applied, some sort of data splitting strategies where the low resolution, so the sort of downsampled versions of the data are stored on SSD servers, and the higher resolution, so the bigger parts of the data set are stored on hard disk based storage clusters, which further drives down the cost because SSDs are really expensive, hard drives are not as expensive, and by taking the stuff that people access most, which is sort of the zoomed out views, if you imagine sort of like a Google Maps view, we take the low resolution stuff, we put it on SSD, and that makes it really fast to access within like 1/100th of a second, give or take a little bit. And then the stuff that's on hard drive is slower, but because you aren't accessing it as much, it doesn't impact the sort of user experience as much. And so what this looks like all put together, and I apologize because the video is like very compressed for some reason. This is a 8 terabyte data set from a one of the Nature papers that I cited in the very beginning of the talk, where this is an 8.8 terabyte whole brain mouse image. And as you zoom around in this interface, this is that Google NeuroGlancer interface that I mentioned. You can see that the image gets higher and higher resolution as it sort of loads in. So this is the CDN running on our server, taking this data, pulling it out from all of our data archives, reconstructing the image and sending it to the user and like all of these different sort of views. So this is XY, ZY, or ZX, ZY, right? And you can see it's relatively performant despite the fact that you're manipulating it like a 9 terabyte data set. So this is it. Oh, this is one that video that wouldn't work earlier, or the same data set as the video that wouldn't work earlier. So this is that the rainbow data set. Here I'm kind of like stress testing it, so I'm kind of jumping around in the Z dimension. So I apologize if the flashing is troublesome. And you can see, you can zoom out, zoom in, and you can see single cell level data, and it loads in relatively quickly. But then you zoom out, and you can see the whole tissue, kind of like a Google Maps. I keep using that analogy. Type of view. So this allows us to, this data set, I know we skipped over it a minute ago, this is about a 10 terabyte data set, and showing you what it looks like with our labs data. So the other piece of this is the neural networks, so the cell reconstructions. So here I'm showing how these two pieces kind of play together. So here the neuron reconstructions are stored in a SQL database that's running alongside the CDN server. And so it's both delivering the images, which are the sort of brain outlines that you see up there. The images are being delivered, but then also all of the 3D models of the neurons are being delivered from the SQL database to the user interface, sort of in not quite real time because they're preloaded, but they load them as these 3D models. So as you sort of zoom through X, Y, and Z, you can see the sort of slice through of these 3D models. But then this is the whole model. And again, this is data from that one Nature paper that I had the citation for a minute ago. So this is kind of showing you how we store and access our data. But then I have all these neuron reconstructions. Everything I've shown you so far is stuff that is from published data sets, right? So for our lab, and the reason I kept this in, even though it's relatively old, is that when I gave the other bioinformatics seminar that I did, 5-6 years ago, whenever it was, I was actually talking about this tool. So InTracer1 is a piece of software that was developed in the lab when I was, like a first-year grad student, that is using Fiji, so that's the thing that you see there on the right, Fiji plus a custom interface to go through and annotate multispectral neuron 3D images to create these models. Now, the issue with this, which it was good for its time, but it has a lot of limitations, including the fact that it is a desktop application. So if you have data in the cloud, it's basically useless for you. It doesn't allow you to access data that's more than tens of gigabytes, which obviously for us is pretty useless. I think the largest data set we ever sort of analyzed fully within InTracer1 is like 100 gigs, which again is not very big in these sorts of scales. And then it also doesn't support like multi-user sort of interaction or like I said, network-based data storage, which at the time was a really big thing on our mind because that paper was published in 2019, 2020 or something, and obviously everyone went home during COVID, and it created a huge pipeline issue for us where we were literally mailing hard drives to collaborators and students who are working at home because they couldn't come into the lab to actually work on our workstations. So we created a piece of software that we call InTracer2, very clever name, which is essentially the same idea as InTracer, but using that Google NeuroGlancer interface instead of Fiji. So instead of, a lot of those limitations that I just mentioned are limitations of Fiji or ImageJ you may have heard of. And so we thought, what if we could bring the same kind of interface into InTracer, or into NeuroGlancer to create InTracer2. So a lot of the videos here are not very great because they are taken on my, or because they were taken on my like Linux laptop where the screen is not very big. This is just to give you a little bit of a feel for what it's doing. So over here on the right-hand side, you have InTracer 2, the actual user interface. It replicates a lot of the menus from InTracer 1. And essentially what's happening here is that this is one data set that's being loaded. It's a little bit hard to see, but I'm clicking on this like purple neuron here, and you can see little, when I click, little dots appear. And as I do this, it is actually pushing, yeah, so the little dot just appeared right there. And I apologize because it is kind of small. And then when I hit a button on my keyboard, it actually passes these coordinates to the server. And then on the raw data, it's actually doing like an A-star path tracing between those 2 points and returning those coordinates back to the InTracer 2 application. So you can start to create these 3D structures as you're zooming in Z without any of the data ever actually leaving the server. So this is here just for your visualization purpose, but none of the computation is actually happening with the data that you're downloading from the server. It's all happening server side. And then as you do this, this is pretty far into an experiment. So we've traced 248 neurons in this specific experiment, and only this purple one that's being worked on right now is being highlighted. But each one of these has like sort of a tree-based structure that represents the morphology of the neuron, which can then be exported to the interface I just showed you. Yeah, so here's just another screenshot of a slightly more complex experiment. So this one, I believe, has about 400 neurons or something that were traced. And the thing we did here is that this data set is, again, not super big. It's like 200 gigabytes. But we essentially selected a region in the middle, and we traced every neuron that projects through that sort of center part. So it creates this sort of like spaghetti ball of neuron interactions that we then rendered with some bioinformatics tools that our lab developed for this purpose in 3D. So it creates this kind of like nice 3D model. But this is what it looks like if you just kind of take a blade and slice through the middle. You can see all of the sort of nightmare of spaghetti that kind of comes in through the center there. So bringing this back to the line confocal sort of context, we have a couple of data sets. This is a fruit fly central brain, basically with the 2 sides of it cut off that we imaged. So this is a couple of terabytes. I think it's 2 terabyte data set. And we went in and selected a bunch of neurons to reconstruct in this specific brain. So here's a couple of them projected down here. And bringing them out, this is what our tracings look like. And what we've done is gone back and identified them in electron microscopy data. So we've mapped this data back to electron microscopy data for the purposes of identifying these cells that we reconstructed. And these are the paired electron microscopy versus our tracing sort of results here. So you'll notice that a lot of ours are these kind of like simpler looking structures because we don't quite have the resolution of an electron microscopy experiment. But the difference is that this experiment took 2 guys, basically a weekend to do, whereas the electron microscopy experiment that we're comparing against was a months-long endeavor with hundreds of co-authors, right? So it starts to identify this whole pipeline as kind of a toolkit where you can interrogate these systems without needing like a dedicated department working on it, right? And sort of study this at a single cell level. Yeah, so that was the end of the sort of more bioinformatics heavy stuff. I think I'm actually technically out of time, so if you need to go, feel free to go. I'm just gonna like glance through these last couple of slides because it goes back to the question that was asked earlier, which is essentially talking about the lossy compression for the image storage. So everything I was showing you was all done with lossless compression. So that's an algorithm that you put an image in and you get out the exact same image, right? So you put in an image, it gives you something that's 3 times smaller. You plug it back into the decompression algorithm, you get back the exact same image. So we've explored basically taking this and applying it to specifically AV1, which is a video compression algorithm. Now, this becomes a little bit more of a complicated discussion because you have all of these variables of how you run the video compression that can sort of impact things. But the key thing is that, depending on the quality, this x-axis is basically quality for all of these graphs. Depending on the quality you choose, it can be, hundreds to thousands of times smaller than your original data, but at the expense of, I guess I didn't include in these plots, at the expense of, quality, but also at the expense of speed. So the higher the quality is, specifically when you're writing your data, it becomes very, very slow because of just how expensive these algorithms are to compute. This is just showing what this looks like, low to high quality, different images. This is one DRAC5 nucleus, and then zooming in. High quality, low quality blurs a lot of stuff out, but this data set is like 100 times smaller than this one. So, it's kind of pick your poison as to how that's important. Inside of our lab, we bought this dedicated server that has 8 16 node machines to experiment with this. I won't really talk about that. We took this big data set I talked about earlier, put it on that server, and in about a day or 2 days worth of time, it went from 100 terabytes to 150 gigabytes in one of our tests using a quality score of 20. So that's 666x smaller. This is just plotting raw data compressed with Z-standard, this data set down here. We implemented on the CDN side so that it can be decompressed, and it's within it's about 150 milliseconds on average, which is still relatively fast for data that is this far compressed and this far processed. And then implementing on the CDN, this is like accessing the compressed data live. So you zoom in and you can see like it takes like a fraction of a second to kind of pop in, but we're accessing the compressed data directly where the compressed data has been stored on an SSD and is being real-time decompressed. So we're able to still keep, while not at full quality, the sort of full resolution of the raw data without the cost of storing 100 terabytes of data. That's the summary. Yeah, and then I have acknowledgements and stuff. So if anyone has any questions, I'd be happy to answer them, but we are already done on time.
Given that we are pretty close to the hour, there is actually a question it looks like online, so I'll quickly read that, sort of long. But that might be the only question we can take. The rest may need to go offline. But so great talk and wonderful work. 2 questions, actually. This reminds me of the thinking microscope that Steve Finkbeiner and Jeremy Lindsley have created at UCSF, which uses reinforcement learning to collect data, perform an analysis, and design the next step of data collection.
Yeah.
Have you explored something like this, perhaps to see where it might be useful to recapture or have additional data collected or a variety of other things for the most time-consuming and challenging parts of a workflow in neuronal segmentation, reconstruction, annotation? And then what would be the feasibility of combining something like this with non-destructive spatial omics methodology out there currently? I would be curious to know the added value that these data would contribute compared to current spatial omics technologies, which are typically not meant for 3D capture and reconstruction.
Sure. So the first question about the microscope, I'm not familiar with that specific paper, but there's one that sounds very similar to that was called a smart scope. And so the idea there was that you basically take an image, it analyzes the image, and it says, oh, there's a cell, and the cell's going this way. So you kind of shift your image over, and you keep following the cell so that you can capture really big things without capturing the whole area. For our approach, we haven't really explored those, mostly because we're usually interested in samples where cells are everywhere. So there's not that much to gain from sort of smartly choosing the area that you're scanning. But for those experiments where there's very few cells, that kind of approach could be useful. We just haven't really looked into it. For the second question, as far as like using it for spatial transcriptomics, so the big challenge there with most spatial transcriptomic methods currently is that they rely on relatively thin samples. So if you think about like Xenium or any of those types of methods, they are actually relying on the RNA sort of diffusing through the sample to a certain extent. And so if you have a really thick sample, which is what you need to make these 3D reconstructions, you're not going to have that RNA diffusion sort of happen. Now, something that the lab has been working on is actually combining the sample tissue preparation methods with like Microscope and some of these like RNA probing based approaches, which isn't really, I mean, I don't know if that falls into the category of transcriptomics, if it's just like RNA staining, but you know. So some of those methods with optimized protocols could be applicable because you can still, as long as your probes can diffuse in the right amount of time and so forth, you could combine it with that. But really the big challenge is that when you're taking something that's, you know, 0.5 millimeter, which sounds really small, but in terms of like tissue histology is relatively thick. When you have something that's that thick, getting anything to diffuse through it in a reasonable amount of time is actually a challenge. And so you start to run into things like that.
Great. Well,